So I also work with people who work with animals, but these animals are a little bit more complex than C. elegans and flies. And I'm going to describe to you this very simple task simply because I'm going to use it during my talk as a way to illustrate the kind of things that we do. So this is a very simple task. It's called a center out task. So imagine you have in front of you a circle surrounded by 8 circles. And in every trial, one of the 8 circles on the outside illuminates. So you go to the center, you wait for one of those to illuminate, then you wait for a go cue, and the interval that you have to wait for the go cue is variable between half a second and a second and a half, varies from trial to trial. When you get the go cue, you have to move this manipulandum that moves the cursor on the screen. You capture the target, you get rewarded, you go back to center for a new trial. Every trial corresponds to a different target, one of the 8 targets chosen at random, and you do many trials per minute. The trials are really short. So what we do in labs that work with monkeys is we record neural activity. This is not fMRI, this is not calcium imaging, this is real spikes. And the way we record this neural activity is by putting multi-electrode arrays like this. This is a 10 by 10 electrode array. It's about 4 millimeters, so the separation between adjacent electrodes is about 400 microns. And you just put it in. It's like, it's almost like putting carpets, really. It's a pneumatic gun that puts it in. And for this particular kind of task, it's put in area in M1 in the arm area, contralateral to the arm that the monkey is moving, so that we can take recorders of the area of motor cortex that is involved in this task. And we get data that looks like this. So this is the type of data that Elad Schneiderman showed you earlier in the week. Every row corresponds to a neuron. The horizontal axis is time. This is about 20 seconds of recording. And during this time, the monkey has made several reaches to one of those targets, so several trials. So this is a typical data. This is also very typical data. And I particularly like to show this because this comes from the lab of John Donahue at Brown. And this was taken from patients that were waiting for epileptic surgery and agreed to have one of these electrodes implanted briefly while the dura was open, waiting for epileptic surgery. It's the same convention as before. Every row is a neuron, every tick is a spike. As I told you before, this is about 20 seconds of data, a bit more, almost 30 seconds of data. And it's 110 neurons. The reason why you record, I'll take your question in a second. The reason why you record more than 100 neurons is because sometimes one electrode can capture several neurons. And by looking, that was your question, and by looking at the shape of the action potential, which is something that I think, Suzanne, you showed us in your talk, different than what you call the neuron individuality or the neuron personality, that every neuron has an identifiable shape for the action potential. Sometimes you can discriminate 2 or 3 neurons that are converging and being captured by a single electron. It's going to be of the order of 100. Could be a little bit less because some electrons might not be properly connected, and can be a little bit more because of these multi-unit signals at given electrodes. So as it says here in the bottom, in this case, we're able to record 110 neurons. M1, this is primary motor cortex, as I showed you. Arm area contralateral to the arm that is moving. And this is HS3, human subject 3. So I particularly like having had access to this data because it shows the person is doing exactly the same center-out task that the monkey is doing in the lab that I showed you before. So this data has allowed us to really try to establish a comparison between the neural signatures of this task in a human primate versus a non-human primate. And this will come back later in my talk. Karina.
Do you have any idea?
I cannot hear you.
Do you have any idea which of these neurons might be inhibitory? Like from the waveform, can you tell?
Waveform?
Yeah, which ones might be inhibitory?
We look at the waveforms, we use the waveforms to do spike sorting. The problem with this kind of method is that it doesn't provide any information about the identity of the neurons except for the waveform. You put this electrode a little bit by force, really in an area. We don't know the neuron types that we're recording. We don't even know whether a given neuron is excitatory or inhibitory. Forget about finer details of the identity. We are not even sure which layer we are recording from within the cortex. And actually, as I'll show you later in my talk, with time, the electrode sinks a little bit. And this electrode stayed in the monkeys for 2-3 years. And it's quite possible that the data that we're recording later is deeper than the area that we are recording earlier. So we don't have all this beautiful information about neuron types or things like that, but we have spikes. So this is very much like the data that Elad showed you earlier in the week. He was also showing every row was a neuron, and the horizontal axis is time, and every tick is a spike emitted by a given neuron. And what he showed you was that you could take a very small time being of the order of a few milliseconds, and then every neuron would either make a spike or not make a spike in that time being. So you can get, for a given time being, a word that is composed of zeros and ones. You put a 0 if there was no spike, a 1 if there was a spike emitted by the neuron at that time, and you get what I would call a Bernoulli description, where at every time being, as you move your time being, you have a vocabulary, you are exploring one of these words that are of length n, and in every side there can be either a 0 or a 1. And how many words of that type do we have? We have of the 2 to the n explicitly. And for instance, for n equals 100, 2 to the n would be larger than 10 to the 30. That's exactly the slide that Elad showed you in his talk. So it would seem from this that you're doomed. You're doomed by the curse of dimensionality. So, all the states here are vertices of a hypercube that lives in a 100-dimensional space, and every vertex is specified by every neuron being either 0 or a 1, and you have 2 to the n such vertices if you have n neurons, and all those are possible states of the network. So you could say, well, how can I possibly classify this? Let me look at neuron types, and let me look at the genome, and let me cluster, because I cannot explore this 100-dimensional space with 10 to the 30 possible states in that 100-dimensional space. So are you doomed by the curse of dimensionality when you look at data like this? So this is the main point that I want to make in my talk, and what I hope you'll get at the end of my talk, is that you are not doomed by the curse of dimensionality. The reason being that although you have 10 to the 30 possible states, the system in its dynamics does not visit all these 10 to the 30 states. And this is not a property of the brain, it's a property of the world. So I want to show you this beautiful example. This is a paper that appeared in iClear in 2023. So what these guys did, they use a very well-established method, which is a method called maximum likelihood estimator by Levina and Bickel. This is a method that doesn't find the nonlinear manifold, but gives you a very good estimate of the dimensionality of the nonlinear manifold in which a given cloud of data lives. And it's a method, I don't know if you remember the old paper by Grasberger and Procaccia. It's a method that says locally, I have a point, I sit in that point, I create an epsilon ball. How many points do I find within that epsilon ball? It grows with the rate, it grows with epsilon. It goes like epsilon to the d, where d is the effective dimensionality in that place where you are. So you essentially keep on putting these epsilon balls, get local estimates of the local dimensionality, and then you have a nice maximum likelihood algorithm that puts all this data together. This algorithm has a parameter that is called k, so these columns are colored by k, and they analyzed a lot of data sets of images. In particularly, the one that I like is they look at ImageNet. Now, ImageNet is a fantastic data set of images that has been used to train every deep learning network that people claim has anything to do with how the visual system is structured or how to do pattern recognition. These images, each one of them is 224 by 224 by 3. These are colored images. So each image lives in a point of 150,000 dimensions when you look at the pixels and you allow the pixels to have a 3-dimensional representation for each color. Okay. So every image is a point in this 150,000 dimensional space, but not every point in this 150,000 dimensional space is an image. actually, if you pick a point at random, it will look like salt and pepper, colored salt and pepper, because you have 3 colors, but it will look to you like random noise. This is not an image. So where do the images live? They live in a low-dimensional manifold. And what is the dimensionality of that low-dimensional manifold? Well, you vary this parameter k between 3 and then 5 and then 10 and then 20. And this is for ImageNet. They also look at CIFAR and other databases that people use. But ImageNet is a more complex one. As you vary k, the dimensionality varies between a little bit less than 30 to about 45. So you are not living in a 150,000 dimensional space. You're living the images. If I believe that ImageNet is in some sense a representation of the world, our brain has evolved by looking at images that live in a 40 or 50 or maybe 100 dimensional manifold. Yeah?
So about that belief of ImageNet representing...
Say it again?
About the belief that you have that ImageNet represents...
Can I hear you?
Sorry, let me scream a little bit. So I assume that this x-axis corresponds to a growth in the amount of data that is being used for training?
The x-axis, every data set is a little bit more complex.
Right.
And so you see an increase in dimensionality, but
But you don't see a signature of saturation, right? Why should I believe that this doesn't continue growing up?
As you do what? You can do...
As you make it more complex.
Well, take movies, do the experiments that they used to do at Princeton with the flies. You put a fly in your hat and you walk around the woods in Princeton, and then you capture all the images that the real fly sees in its real environment and do that statistics on the images that you get. If that's a natural word for a fly.
But what if I take woods in Norway, in Italy?
Okay. You know what? The point that I'm trying to make, maybe it will not be 50, maybe it will be 100, maybe it will be 1,000. It will not be 150,000.
Okay.
That's the point that I'm trying to make, okay? The point that I'm trying to make is that the world in which we live, all the inputs that we get, that we're exposed to the visual, the sensory, all the things that come in, live in low-dimensional manifolds within the space of all possible. Images, all possible. Sounds, all possible. That's the point. It's true that if you could make this more complex, eventually, if I have to make a bet, I bet that it will saturate. And it will saturate at not a very big number. And I will bet under 100. Yeah, but it's a bet. It's not a mathematical theorem.
This might be beyond the scope of your talk, but do you happen to know if this analysis was done in the pixel space or in like a latent space of some sort?
This is an in-pixel space.
Really? Okay.
The dimensionality reduction is an in-pixel space.
I would have guessed the kind of nearest neighbors between different images in pixel space was
It's
sufficiently
an Euclidean
meaningless.
distance in pixel space. It's a most brute force method that you could imagine, but it's a smart method. You should look at this. This Levy-Nabikal method is a nice method. So now I want to go back to this kind of data and look at it from that point of view. Not look at it from the point of view that this lives in a 10 to the 30 dimensional space, but look at the dimensionality of the states that are actually visited during behavior, during life, during an animal doing something. And so is it very high dimensional? Do I have to still worry about that? And what I'm going to do is move a little bit away from this Bernoulli description by allowing for slightly different time beams. So let me look how I want you to think of this. So how are we going to describe this? I'm going to take a Poisson point of view. I take n neurons. The activity is observed during a time t. I'm going to use an interval of size delta, so I'm going to use a discrete time that I call little k. K varies between 1 and capital K, where capital K is just t over delta. So I'm measuring times in units of delta, and I'm counting beans instead of thinking time as a discrete variable. And for every bean, for every neuron, I count the number of spikes emitted by neuron I at bean K, which will be not just 0, 1, but could be 0, 1, 2, 3, 4. It's a counting variable now. And we'll go bigger and bigger in the allowed number of counts as I make my delta larger and larger. So I'm expanding a little bit away from the 0, 1 representation. So I do that, and I can create a data matrix that has essentially the same structure as the data. Every row is a neuron. Every column is one of the delta of the time beams of size delta. And in every beam and every row, I put the number of spikes emitted by that neuron in that particular beam. So one thing to remember of this type of method is that we are very proud because if I use these electrode arrays, I can measure of 100 neurons. If we use neuropixels, we can measure a few thousand neurons. But still, I want you to remember that when you do this, to grab your coffee, of the order of 1 million neurons in your primary motor cortex are modulated by that task. So 100, 1,000 sounds brilliant compared to one at a time, as used to be the case not so long ago. In the 80s, the lab of Georgopoulos actually did these measurements where it was a neuron at a time, and there were very important discoveries about direction selectivity. But still, it's a very, very gross subsample. So what I want you to keep in mind is the following. There's a real neural space, which is very high dimensional, and the data is not the real neural space, it's an empirical neural space, which is strongly subsampled from the actual neural space. So there is already a linear projection gone from the real neural space to the empirical neural space that we have access to. So the empirical neural space, I'm going to call the dimensionality capital D, where capital D is simply what people at Janelia have called the ambient dimension. It's just simply the number of neurons that we are recording from. Okay, so I go back to this simple central task. I'll show you this data that I showed you before. And here, during these 20-something seconds, 3 reaches have been made. One reaches to the yellow target, one reaches to the red target, one reaches to the blue target. So what we're going to do is use a very, very wide time interval of 300 milliseconds, which is centered around the go queue. 100 milliseconds before the go, 150 milliseconds before the go queue, and 150 milliseconds after the go queue. So there's going to be only 1 point per trial, essentially. And the point is going to live in my empirical neural space, and the coordinates will be the number of spikes emitted by each one of these neurons during that particular time interval. So I can put all these points in a space of dimensionality equal to 80-something in this case, which is the number of neurons that I'm recording. And then I can look at this cloud of points and ask, is the position of that point predictive of the target in that particular trial? So can I look at the cloud of points and color them according to the corresponding target, and does this color agree with the actual target that was the target for that particular reach? Well, the answer is if you put them in an 80-dimensional space or 100-dimensional space and do random projections, you will not see any structure. But you have to be a little bit smart, not very smart, linearly smart in this case. You can do a PCA or factors analysis. And we were doing it and we had it, but Krishna Shenoy lab came ahead. And my student was very distraught, but I tried to console him by telling him that if we're being scooped by Krishna Shenoy, it means that we're asking good questions because we are on the right track. So this is what they did. They did a 3D plot. Neural mode 1, neural mode 2, neural mode 3 means principal component 1, principal component 2, principal component 3 as they come out from factors analysis. So just a little bit more sophisticated than vanilla PCA. You do that for all your points. Every point here is one trial. You do that for all your data, and then you color them. This is not supervised. This is totally unsupervised. You put the data, and then you color them afterwards because you know to which target that trial corresponded. And when you do that, you see 2 things. First of all, you see that your data is clustered. I have said nothing about connectivity. I have said nothing about genome. I have said nothing about neural types. I just want to show you that these features that we have been discussing appear when you do very different types of analysis than the ones that we have been discussing. And what blew my mind the first time I saw this, not only that there were 8 clusters, each corresponding to one of the targets because you can decode, but look at the order. This is the way the targets are ordered in real space. You go from orange, you go from purple to red to orange to yellow, and here you go from purple to red to orange to yellow. So this totally abstract representation of the task in primary motor cortex is capturing something about the external, the physical organization of the targets. I truly wasn't expecting that, because this doesn't even reflect how you have to control your plant, because the way you move your muscles to go this way is very different from the way you move your muscles to go this way when you have to span the circle. So this is just an elbow joint. This requires moving the shoulder joint. So if you have to think of it in terms of neural instructions to the plant that will make it possible to make these reaches, it's just simply amazing that this happens. Well, this low dimensionality is a persistent feature of cortex. This is the second important message that I want you to take home. It appears everywhere in cortex. There is a very beautiful paper from Cunningham and Yu, where they said that they have this very large neural network. I have relatively few electrodes. In this case, I'm just recording neuron 1, neuron 2, neuron 3. These are the spikes that I observe, and now I put my beam and I move it. Every time the beam is put somewhere, I have a point in this empirical neural space at 1 and 2 and 3. And now I ask myself, as time passes, what kind of trajectory do these points describe in the empirical neural space? And what is observed, and again, this is not a mathematical theorem, it's a very, very strongly supported empirical observation, is that these trajectories, first of all, they are confined. They don't explore the whole space. But they could, in principle, be confined to a cigar that has the same dimensionality as the space, right? It could be exploring a reduced area, but of the full dimensionality. Well, that's not what happens. These trajectories live in low-dimensional manifolds within the neural space, even within the relatively low-dimensional empirical neural space that we are recording. So this goes back a little bit to your question, Sandro. When we look at this very simple motor task, we essentially find 2 or 3 dimensions, right? Now we build a cage and we let the monkey move around the cage, and the cage is a rich environment. So sometimes the monkey is scratching himself, sometimes he's grabbing treats, sometimes he's hanging from somewhere. And we record during 2 or 3 hours, and we ask ourselves, what is the dimensionality that we get? So we don't get 3, we get 40. Okay, we get still much, and it saturates as we bring more and more neurons into the analysis. So it's not that we get 40 because we're only recording from 100 neurons. Now, this is not exactly behavior in the wild, but it's much more closer to behavior in the wild than this very controlled, centered out experiment in the lab. So this is the intuition that goes behind the answer that I gave you earlier.
Yeah, I think for motion it makes sense because
for motor it makes sense because you're still controlling a plant which is low dimensional. You have so many of the order of 100 joints in your body and muscles that you need to control in order to control the joint angles. So why would you need 1 million signals to control that? But what I try to convince you with my first example that comes from the machine learning literature is that this is also true of the sensory signals, because the sensory signals that the world gives to us are intrinsically low dimensional also. Yeah.
So how many of those dimensions, since you're recording from a multi-electrode array, you're gonna have like correlated activity?
Yeah,
right?
It's because of the correlations that the dimensionality is effectively low.
But it's not because of the neurons fire together, but it's because of the recording, because you're sampling from a very small area.
I'm recording single spikes. The correlations that I see are not an artifact of my recording. This is not calcium. It's an extracellular recording of single neurons, individual neurons that have been properly spike sorted. So that is not an artifact of that. So now, having made this observation of this ubiquity of the low-dimensional manifolds, there are many questions that arise. The first question that arises, there was a question? Oh, sorry, Susanna, I didn't see you. You have to yell at me.
I also want you to get through to your talk, but the picture on the slide before, when you showed these 8 clusters, did I understand correctly that it is...
I cannot hear you, I'm so sorry.
So can you go back to one slide?
Yep. That we can do. There?
Where you have the 8 clusters.
Say it again?
Where you have the 8 clusters, right?
The ring and
The ring one.
The one with the 8 different colors.
Oh, in the 8 dimensions you see it. I will give you, in that representation, I was just showing you 1 point per trial. But in a little while, I'll show you trajectories for those tasks. And they still live in 3 dimensions, but you see how beautiful the trajectories are. They look like the petals of a flower. They are really amazing. You just wait a few slides, we'll get there. Okay, so theorists. What do the theories do? The first question that the theories ask is, are these low-dimension manifolds flat or curved? Can I access them using linear methods, or do I need non-linear methods? That's the first big question. And there is a beautiful recent paper by Merhat Sayeri and Zordan Ostojic, where they discuss this notion of intrinsic dimensions versus embedding dimensions. So they say, okay, look, if I take my shoelace and I make an arc, the intrinsic dimension is 1, but I need at least 2 Euclidean dimensions to see it. However, if I pinch it a little bit, I will need at least 1 more dimension. So the embedding dimension can increase as I add more kinks to it, but the intrinsic dimension is always 1. And then they say, okay, if you live on the surface of a sphere like we do, the intrinsic dimension of your world is 2, but you need at least 3 dimensions in order to see it within an Euclidean space. So they distinguish this notion of intrinsic dimensions versus embedding dimension, and they have a very nice discussion of what it implies from the point of view of the nonlinearity of the, and you see it here, that as I increase the number of kinks, let's say, this is living in a 100-dimensional space. So when in 2 dimensions, there are 98 other dimensions in which I could put kinks. And as I involve kinks in more and more of those dimensions, this ring becomes more and more nonlinear, and the embedding dimension will increase, but the intrinsic dimension is always 1. So the ratio or the difference between these 2 dimensions is, in some sense, a signature of the degree of nonlinearity of the manifold. Not surprisingly, and I don't want to spend any time doing this, these latent variables can be used as a generative model. So the activation of these low-dimensional neural modes, which could be linear or nonlinear, from that you can map back to the neural space and extract from that the activation of the individual neurons. So what we argue from a functional point of view in the brain is that what the brain activates is not individual neurons, but neural modes. A particular collective pattern of activations with relative strengths, and a given neural mode could be activated more or less, but the neural mode is a direction in the neural space. It's not neuron 1, neuron 2, neuron 3, but it's a direction that involves many of those neurons, and it's a particular collective pattern of activity. So we know a lot about these latent variables. We know that they represent collective patterns of population activity. They provide a generative model for the activity of individual neurons. They are ubiquitous. They appear, since in the last 10 years, they have been reported everywhere, in sensory cortices, motor cortices, prefrontal, parietal, even in hippocampus, where some years ago, the hippocampus experts believed that I was seeing this because I was working in cortex, but the hippocampus has such a detailed geometry, and it will never happen in hippocampus, and it happens in hippocampus. There are now papers that show that, from the same people that told me that it will never happen in hippocampus, so it was great. So let me concentrate on this point, on the prediction of behavior, because in the beautiful talk by Srini, we saw a lot of how he could use these things to make predictions, the detailed knowledge of the connectome to actually make predictions. So I want to show you a beautiful picture that comes from Juan Gallego, who was at our lab at that time and now is at Imperial College London. And so this is the number of latent variables, and the black curve is the variance accounted for in a standard PCA. So if I ask you, how many latent variables should you keep? Well, you say, well, if I want to account for 90% of the variance, I need like 35 neural modes or something like that. But now I'm going to construct a model that predicts muscle activity that in our lab is measured, in the lab of Lee Miller, is measured simultaneously with the neural recordings. So each one of these funny acronyms here corresponds to a different muscle in the arm and in the hand. The circles at the end show you our ability to predict the activity of that muscle during a reach when all the neurons that are being recorded from are being used as predictors. Okay. You see, it's not very good. The reason being that muscles are hard to predict, particularly when they are measured extracutaneously. These are not measurements that come with needles going into the muscle, but they are a little bit like when you take your blood pressure, essentially. It's a cuff, an external cuff. So these are not very good signals to predict. So as you see, the quality of prediction is not fantastic. It's, it's between 0.5 to 0.7. But the point is that as I increase the number of neural modes that I'm using as predictors, as inputs to my decoder, it very quickly saturates. And by the time I have captured 10 neural modes, I have as much information as what the muscles are going to do. I can extract as much information as if I had used all the neurons or all the modes, which is just a linear transformation away from the neurons. So this is very important because it says it's not just the variance accounted for, and it goes back to this paper, David, that you and I like a lot about communication subspaces. So the variance accounted for might require me to look at a 30-dimensional subspace if I want to see most of the activity in this area. But if I ask how many dimensions do I need to capture in order to understand the muscle activation, it is of the order 10, not of order 30. So there's clearly a communication subspace through which the information is transferred to the muscle. And just to show you that I can also do worms and flies and things like that, it's not me, actually. I just want to show you another paper by Breman and Prödke, where they look at C. elegans. And they constructed this beautiful manifold. This is, again, a form of PCA. It's the mixed PCA. It's a very elegant technique that comes from the Champalimaud lab and Byron Yu. And so they have this very elegant manifold. And what is very interesting, first of all, this is all neurons in a worm. And this is by registering across worms all the neurons that they can, in their experiment, identify that it was the same neuron in different worms, right? So it's a bit fewer neurons. But look at the robustness of the shape of this manifold. Moreover, they are looking at different behaviors, forward versus backward locomotion, reverse to one direction, reverse to the other one, a turn in the dorsal or ventral direction. And along that manifold, there are regions that correspond to each one of the movements. So by knowing where you are on the manifold, by looking at the neural activity, tracing this manifold, and know where you are in the manifold, you know what the worm is doing without looking at the worm. You can predict which one of these modes of behavior from the manifold. Okay, so again, we don't need the full neural space. We just need the manifold that is controlling that behavior. I'm personally very interested in behavior, so this is, to me, it's a very important observation. I want to talk. How much more time do I have?
15 minutes. 15.
15, perfect. I want to close by showing you about alignment, because that's the other thing that we keep on talking about that you can do if you have more information about the connectivity of the network. So this is the same center of task that I told you before. The monkey has been doing it for about 3 years almost, or more than 2 years, 800 days. And these blobs correspond to the times when the monkey has been in the lab doing this task. And what you can see, these are the trajectories to the 8 targets. So you can see it's very stereotyped. Once it learns the task, it's like when you see a good tennis server, right? You can know who it is by the way it's served. They have a very stereotyped, distinct signature. So these trajectories are very stereotyped over 700 days. If you look at one of the directions, like up and to the right, and you look at the x and y components of velocity, they are also, for that particular reach in that direction, they are also very stereotyped and conserved across days. These are the correlations of the x and y components of velocity across 700 something days. You see they are all very close to 1. So it's a very stereotyped task. So we conjectured if the task is very stereotyped in the execution, there has to be a neural representation that is also very stable, right? The problem is that we cannot compare the neurons recorded on day 1 with the neurons recorded on day 700 because the electrode moved, it's not recording from the same neurons, some of the electrodes got short circuit because of scar tissue, so it's not the same set of neurons. So in order to check this hypothesis that the neural activity was also preserved, we need to correct for the fact that our empirical neural space is not the same across so many days. And so this was our picture. Our picture is that there is a mother neural space which has many, many dimensions. That's the one at the top left. In that mother neural space, there is a neural manifold that corresponds to this task, and there are trajectories that correspond to this task. So the first thing we do is we project into our empirical neural space. And that projection is not the same on day 1 than on day n, because we're measuring different units. But that projection is itself a linear operation. Then in each one of these neural spaces, we're going to do dimensionality reduction using PCA to find a hyperplane that contains most of the trajectories, which is again a linear operation. So we do 2 linear operations to get to our trajectories in day 1, and 2 linear operations to get to the trajectories that we see on day n. And so we ask ourselves, shouldn't it be possible to align them using a linear method? So we started with canonical correlation analysis. Like we say, it's the same object, right? It's the same object. I'm just looking at it from different perspectives, projecting it linearly. Is there any way to extract the original object? And this will take me to the trajectories that I wanted to show. So canonical correlation analysis is a very simple. Don't ever neglect your linear methods. This is what I always tell my students. We are always going to start linear. See how far it takes us and see how it breaks. And from that, we'll get some intuition of how to go nonlinear, from how the linear method broke. Never start by doing a zebra, or always start by doing something linear and seeing what you learn from that. So this is a linear alignment. So you do a linear transformation in your hyperplane at day 1, and a linear transformation in your hyperplane at day n. And these are not just rotations, they are rotations and heterogeneous scalings, different scalings along different axes, essentially. It's something that you could do a QR decomposition on, to be more explicit. And we learned that from a very nice paper from Bahab Jordan. This comes from the machine learning literature, and we do it. So here I'm showing you data from another monkey that we recorded for 45 days. So you see in day 1, the 3-dimensional trajectories. And now what I've done is I've averaged over the trials to a given target. So I'm only showing you 8 trajectories, 1 for each of the targets. I'm showing them to you on the 3-dimensional hyperplane that results from looking at the data of day 1, and the 3-dimensional hyperplane that corresponds to looking at the data in day 32. I do the alignment, so a linear transformation on day 1, different from the linear transformation 2, that surges to increase the correlations. This is called canonical correlation analysis. The goal is to increase the correlations between the transformed trajectories and the transformed trajectories. So first of all, look. They look very similar after you've done this very simple linear transformation. They look very elegant because they look like petals. Now I have a trajectory. I go to the target and come back. And if you look at the order of these trajectories, they are ordered again in the same spatial organization as the targets. Hold on a second. Does that answer? Is that what you wanted to see?
I wanted to see when after the decision was taken where to go, you can distinguish the clusters.
Yeah. So what you need essentially is a better time resolution. Here we were looking at the time scale of 5 to 10 milliseconds per beam, as opposed to 300 milliseconds, which only gave us 1 point per trajectory.
Is the linear transformation that relates across days like close to being an isometry?
It's close to being a?
Isometry, like a pure rotation without scaling?
No, the scaling is important because remember that I'm compensating for the fact that I'm looking at different neurons with different gains and I'm looking at the hyperplane in one neural space, and the other hyperplane is in a slightly different neural space. Some neurons are shared, but there are neurons here that are not here, and neurons here that are not here. So I'm not answering your question.
Yeah, I feel like people think about like neural trajectories as being like meaningful up to like rotations, but not necessarily
because you're thinking of the same neurons. I think if you're thinking of the same neurons, that would be true. But here I'm also trying to compensate for the fact that I have some non-shared neurons, and they can have really different gains because they can have very different mean firing rates. So if they have a different lambda, they have a different mean, a different variance. So the scaling becomes non-isotropic. Okay. I don't want to tell you anything about this. I want to say something interesting about alignment. So I told you that we could predict muscle activity from the neural activity. So we have neural activity, we have a decoder, we predict muscle activity. And our experiment allows us to construct the decoder because we simultaneously measure the neural activity using these electrode arrays and the muscle activity using this kind of cuff on the arm. So that's what we do. We have this monkey that we call the source monkey. We have the firing rates. We do a dimensionality reduction. We extract latent signals. And from the latent variables, we have a simple decoder that predicts the envelope of the EMGs. This is just a linear transformation to begin with. So now I have two monkeys. And I ask myself, wow, this guy said this C. elegans can be mapped onto this C. elegans. Okay, you say it's only 300-something neurons. The neurons can be labeled. This is a monkey, a cortex. I don't know which neuron here corresponds to which neuron there. Can I align? And the answer is yes. I can align if I try to align the latent variables. So what we do is an experiment like this. We take, these are again, I'm showing you trajectories to the 8 targets, and the thick lines are the average trajectory, again, color-coded. So I have what I call the source monkey and a target monkey. So I do the canonical correlation analysis, and this is how close I can bring them. But now what I can do is, I have the source monkey in the lab for a long, long time. I measure neurons and muscles for a long time. I have a very good decoder for the source monkey, but I don't have such a good decoder for the target monkey. So what I'm going to do is take the target monkey, do the canonical correlation analysis to get to the shared image, invert it this way to get to the source, and use that as input to my decoder. So target monkey goes to the shared representation, which gets inverted using the canonical correlation analysis. Sorry, I messed this up, of the source monkey, inverted, sorry. I have to change that. So that's what we do. And we ask, how good is the prediction of EMGs? Using the decoder that I constructed for the source monkey. And it's very good. So this is what we're doing. We're getting the monkey, what I call the source monkey. We get the latent signals. We use the canonical correlation analysis to go to the common variables. We invert it for the source monkey. We go back to the source monkey, and we input that to the decoder that was constructed for the source monkey. And you can see here for the 8 targets, again, these funny acronyms correspond to different muscles in the arm and in the hand. And you can see what is the correlation that I get from how good performance I get from itself versus how good performance I get when I use this transfer, and you can see that the alignment is quite good. Now you can say, okay, just why do you care about this? Just keep the monkey longer in time. This is a blown up with only 4 muscles, the 4 more important muscles, for you to see how good or bad the prediction is once I do this transfer. And you could say, okay, why is this at all important? And the reason why it's important is because now I can align a human with an injury for which I cannot take brain and muscle data, because if the human has a spinal cord injury, cannot move his arm. But I have a very good monkey decoder, and now I can map the neural activity of the human into the neural activity of the monkey, use the monkey decoder to predict muscle activation, and use that signal to actually activate, thank you, the the muscles and the person can move the arm, although there are no signals going directly from his brain to his arm. So I can not only align across non-human primates, I can align a non-human primate to a human primate. And now I have my homework message, which has to do with what art theory is supposed to do. So there is a very, very beautiful paper from Francesca Mastrogiuseppe and Søren Ostojic, beautiful paper, where they had this lovely idea of using low-rank connectivity matrices to explain why this nonlinear dynamics is so prevalent. How does this nonlinear dynamics arise? Why do we see it everywhere? What is the mechanism underlying it? So they use a very, very simple model, the usual dynamical model, where, I don't have to explain this, we have seen it many times, but the Jij now was the outer product of two vectors. So it's a little bit similar, David, to what you're using in your poster to do the connection across regions. So to make it low dimensional in some sense. So in these cases, the matrix Jij is a tensor of rank 1 because it's made out of the outer product of Mi as a column vector and Nj as a row vector. And there is also a strength G, that is the trick that people always use to look at chaotic dynamics. And I want just to show you this figure of this paper. I'm not going to explain it. It's figure 1 on their paper, just to show you that if you construct a recurrent network with the Jijs that come from this outer product, as you change the degree of overlap between these two vectors and you change the strength J of the gain of the synaptic strength, you get the usual chaotic regime, but you also get a lot of structure. And the most important thing is, as you would expect, you get low-dimensional dynamics. So low-rank connectivity is sufficient, or the low-rank connectivity of the connectivity matrix is sufficient to get low-dimensional dynamics. But is it necessary? And there is another lovely recent paper in the archives from Søren Ostojic and Stefano Fusi, where they argue that this structure, the geometry and the modularity in neural activity, implies a structure in the connectivity. And the question is, how are these two things related? To which extent the prevalent observation of low-dimensional dynamics tells us something? Which constraints does it place on the connectivity? Can we say something before, I'm not going to get a genome for M1 in any near future, right? So a connectome, sorry, for M1. So what can I say about the statistical properties of the connectivity in M1 from the fact that or the cortex in general, from the fact that we see everywhere these low-dimensional dynamics? And this is my last slide. We talk about neurons. So visual stimulus go to visual neurons, and then they go to motor neurons, we go to muscle activation, we go to body motion. On my point of view on this is that there are low-dimensional representations at every one of these levels. That the visual neurons and the motor neurons can be described by the neural modes. That the muscle activations are also very strong correlations and can be described on the basis of a very few collective degrees of freedom that are called muscle modes. That's a very old notion called muscle synergies. And then, as you know from studies, for instance, on the fly, many of the patterns of behavior can also be decomposed in a small vocabulary, which is the vocabulary of behavioral modes. So my preference, this is my last slide, is, I shouldn't call it a preference, I don't know. I think that going from neural modes to muscle modes to behavioral modes is a productive way of thinking about what we really care, which is how neural activity gets transformed into behavior. And the part I told you about work of other people that was relevant to the argument that I was trying to make, but the part that is our work, Lee Miller runs the Monkey Lab, where the data is obtained. Juan Gallego and Matthew Perrich were a postdoc and a graduate student in the lab, co-advised by Lee and myself at the time we wrote our 2017 paper on this, and then more recent analysis trying to understand when the manifolds are linear versus nonlinear have involved Fabio Rizzogli, who is still a postdoc at the lab, and Ege Altan, who just got his PhD. Thank you.